Hello students, we're in Anatomy and Physiology 1 and looking at the muscular system part 2. Last time we were together we talked about the sarcomere and the sections of the sarcomeres, the different parts, the I band, A band, H zone, Z line, and M line. And we looked at the thick protein filament called myosin and with the myosin heads, and the actin with troponin and tropomyosin. But let's look a little bit at regulation of contraction, and then we'll move on and talk about how those sliding filaments work for muscle contraction. Muscles work by activating these smaller units called motor units. A single skeletal muscle or any muscles will have many motor units. Okay, so to regulate the strength of the muscle contraction, we look at the single motor unit and how they work together with other motor units to regulate that contraction. A motor unit is defined as a motor neuron and the muscle fibers it is connected to. Okay, let's look at this picture here for just a minute. We have a motor neuron here, this myelinated motor neuron, and then this motor neuron will branch, and that's called a collateral. And there's another collateral. There'll be many branches or collaterals for this one motor unit or motor neuron for this one motor unit. And then near the end of that axon are these telodendria, and with synaptic knobs that are attached to the muscle fibers, and this myofibril, this bundle of myofibrils called a motor unit. And where that synaptic knob attaches on that motor unit is called a neuromuscular junction. Portions of the skeleton muscle fibers, those neurofibrils there, and that's the part of that motor unit. Again, there are many motor units in a muscle. And as we see, this is a motor neuron, so the muscles are stimulated by the nervous system. And when it is stimulated and you have contraction, that is also called an action potential. So an action potential equals muscle contraction for a muscle. When we get to the nervous system, an action potential of a neuron is when you have a nerve impulse. And you could have action potential with a neuron or all the nerve fibers in that nerve, and when they fire, that nerve impulse is called action potential. Here, when the stimulation is propagated down the motor neuron to the collaterals, to the telodendria, to the synaptic knob, and then to the sarcolemma, over to the sarcoplasmic reticulum and the transverse tubules, it leads to stimulation and an action potential. And how does this happen? It's called the sliding filament theory. Okay, a motor unit is a single muscle fiber. In that motor unit, a single muscle fiber works in an all or none fashion. So you have one motor neuron that branches off in these collaterals and will stimulate that entire motor neuron, so that motor neuron works in an all or none fashion. Now again, there are many motor units in a muscle, so to get a slight contraction, only a few of those motor units are recruited, and then to get a strong contraction, like a tetanic contraction or a Charlie horse, all of these motor units would be stimulated and contracted. Okay, not all or none. If it gets everything, all the things that have to line up for a muscle contraction to happen, if it happens, then that whole motor unit responds in an all or nothing fashion. Either it doesn't work together and contract, or it does. Once an action potential reaches the muscle fiber, all the sarcomeres and all the myofibrils contract. But muscles are organs, and they can have a graded response. And a graded response, how much the muscle contracts, each muscle is served by at least one motor neuron. Now the muscle itself, at least one motor nerve. What's a nerve? It's a bundle of these axons, okay, of a motor nerve. So this is just one very small portion of that motor nerve. An axon is from a single motor neuron. A single motor neuron will have only one axon. A motor nerve has many, many motor neurons. Okay, so a graded response would be like several of these motor units activated and beginning the contraction when other motor units do not. That would be a graded response. The motor nerve is a bundle of axons from the peripheral nervous system. Now the brain and spinal cord, the axons, neurons that are within the brain and spinal cord is called the central nervous system. All those nerves that come off of the spinal cord, the brain and the spinal cord is called the peripheral nervous system or peripheral nerves. And so these muscles are innervated by peripheral nerves that innervate that muscle, and it's broken down into these different axons of that peripheral nerve innervating motor units. So there can be hundreds of axons. Each axon ending forms a neuromuscular junction with one muscle fiber. The muscle nerve fires. When this motor unit fires, all the muscle fibers in it will contract. There could be several hundred of those muscle fibers in that all or none response. Now, neuromuscular junction. Now, we had a picture of a neuromuscular junction in lab, or when we looked at the model, the pink colored model, we saw a neuromuscular junction on the side of that model. The neuromuscular junction is where the site where the axon and the muscle fibers communicate. It is the neuron will be a motor neuron of the peripheral nervous system. There's a myelin sheath covering that neuron, and by the way, we're not to the nervous system yet, but that myelin allows for a really fast nerve impulse to be propagated over that axon to the synaptic knob. This whole thing here is called the axon terminal or telodendria, and then you have the synaptic knob, and there at the synaptic knob is enlargement here. You have the motor end plate. This area here is called the motor end plate. This area here, the synaptic cleft. Okay, this is the synaptic bulb or synaptic knob, and here's the motor end plate. That is that undulated surface of the sarcolemma on that muscle at that neuromuscular junction, the motor end plate. Now, we mentioned before that this undulation of tissue allows for more surface area, because on the surface area of this tissue, on the surface of this tissue are going to be receptors that are stimulated by the neurotransmitters that are in these synaptic vesicles. Okay, in between the axon and the motor end plate, the muscle and the axon don't truly touch. There is a cleft here, and in that cleft you have like a bioelectrical gel that allows for the transport or the movement of that neurotransmitter across that cleft to the receptors on the motor end plate. Now, the neurotransmitters are chemicals that cause things to happen here, or it can be just the opposite, keeps things from happening here. So there are two basic types of neurotransmitters, an excitatory or an inhibitory. Excitatory, something's going to happen. Okay, an inhibitory keeps things from happening here. Okay, and we'll look more at that a little bit later on. Okay, let me go through this again with you. This is a neuromuscular junction where you have the nerve stimulus, and the muscle cells are stimulated by a motor neuron of the somatic or voluntary division of the nervous system, and any output from the central nervous system neuron is a nerve cell capable of sending messages and transmitting messages from the central nervous system to the muscles. You're going to have the axon ending, the distal part of the neuron, the area of the neuromuscular junction, and this is that whole area here is the neuromuscular junction. With each muscle fiber, there's one per fiber, muscle junction per fiber. If you've ever seen a TENS unit, that's a unit a person might wear for pain relief, or an EKG lead has many leads from one cable, and that's pretty similar to what happens here in the telodendria, where you have collaterals and telodendria of that motor neuron. The synaptic cleft, we just talked about that. A space that separates the axon endings from the muscle fibers. It's filled with a gel-like substance. Then the synaptic vesicles. These structures here are the synaptic vesicles. In motor neurons, axon terminals, you're going to see synaptic vesicles. And within that synaptic vesicles are going to be neurotransmitters. For muscle contraction, a neurotransmitter is, the main neurotransmitter is acetylcholine. And in this part of the picture you'll see ACH there. That is the abbreviation for acetylcholine. Neuron transmitter. Acetylcholine is excitatory. Inhibitory or excitatory are two different classes of neurotransmitter. Acetylcholine is excitatory. So acetylcholine, if all the dominoes are set up and ready for a muscle contraction, when acetylcholine is released, things begin to happen to cause that muscle to contract. And acetylcholine is the neurotransmitter chemical allowing the neurons to communicate with each other, causing muscle contraction or a neuron to fire. Okay, so we talked about neurotransmitters being excitatory or inhibitory. Inhibitory would block the nerve impulse, and excitatory would take you closer to a nerve impulse. Now, stimulation for a contraction, acetylcholine is that excitatory neurotransmitter that you have to have plenty of for those motor units to contract. The nerve impulse causes release of acetylcholine from the synaptic vesicle. Now, let me say this. We have talked before about calcium. For these synaptic vesicles to open and release by exocytosis, by the way, release their acetylcholine, you must have an influx of calcium into that synaptic knob, and that influx of calcium will bind to that neurotransmitter synaptic vesicle that stores that neurotransmitter, the synaptic vesicle. It'll bind to the synaptic vesicle, and then that synaptic vesicle will release its neurotransmitter by exocytosis in this case. Now, there's many different neurotransmitters, but for muscle contraction we're going to focus on acetylcholine, and so it allows for the release of acetylcholine from that synaptic vesicle. And the binding of the acetylcholine to the receptors, remember I just said that on that motor end plate, that undulated for increased surface area motor end plate, there are going to be receptors that will bind to or allow the binding of acetylcholine to the membrane, that sarcolemma of that motor end plate. It generates a muscle impulse. So you have the influx of acetylcholine through these acetylcholine receptors. Now, the receptors, acetylcholine binds to it, and it allows for sodium to rush in and change the polarity of this membrane so that you can have an action potential. The muscle impulse actually reaches the sarcoplasmic reticulum, where things continue to happen, and it can lead up to an action potential.
Potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
And there you have the synaptic vesicles with their acetylcholine ready for the next influx of calcium ions so that these synaptic vesicles can release the acetylcholine. Now, a lot of things have to happen for this membrane to be ready for another impulse, and the whole process starts over again. Okay, before we start or continue talking about muscle contraction, let's go over these parts again so you'll be familiar with them as we go through these steps of what happens for a muscle to contract. This is the myosin with the myosin heads, okay? And this is that thin actin protein, that thin protein filament actin. And bound to that thin protein actin is tropomyosin, like that thread that encircles that thicker rope. Okay, the protein, a thinner protein filament called tropomyosin, and then a globular protein called troponin. Okay, so there's the globular protein troponin, there's tropomyosin. We have a model upstairs in the lab that shows you these things. There's the head of the myosin. So the thick protein filament myosin with its head. You'll notice that the heads have attached to them ADP plus a phosphate. Remember that ATP is adenine triphosphate. This is diphosphate. So we know that ATPs are our currency for energy production. So when you split that ATP, when you have decomposition of ATP, catabolism, that ATP becomes ADP and phosphate. And when you split that ATP, there is energy involved, and that energy is stored in this myosin head. So it is like cocked and ready to go for a sliding filament and muscle contraction. The muscle impulse causes the sarcoplasmic reticulum to release calcium ions into the cytosol. Now we saw that in the last slide where we had the neuromuscular junction, the influx of sodiums, and sodiums rushed in, and it changed the polarity of the membrane of that muscle fiber. And when it changed the polarity of the membrane, it caused the release of calcium ions into the cytosol. The calcium ions released into the cytosol will then bind to the troponin, and when it binds to the troponin, it changes the shape of the troponin, which changes the shape with the help of the tropomyosin, changes the shape of this actin molecule, and the binding sites are now exposed. Now in this previous drawing, the binding sites are covered up. You haven't had the influx of calcium to bind with the troponin, so nothing's going to happen. You are not going to have muscle contraction. When you don't have enough calcium, this will be the results. Enough calcium in your body, blood calcium. Okay, the influx of calcium, it binds with the troponin, changes the shape of this actin filament. With the help of the tropomyosin, it sort of twists open that actin filament to allow the binding sites on that actin filament to be exposed. These have to be exposed before you can have the binding of the myosin head to the actin. And that's exactly what the script says. The calcium binds to the troponin and changes its conformation or shape. The position of the tropomyosin is altered to reveal the binding sites on this actin, and that you will have the binding of the actin and the myosin, the head of the myosin with the binding sites on the actin. And then what happens next is called a stroke, a power stroke. And all of this is called the sliding filament theory. the cross bridge pulls this is cross bridge, when the actin and myosin head bind and you have a power stroke. This is called cross bridging. When they bind, then you have the power stroke. The ADP and phosphate is released from the myosin head, and then you have an influx of ATP. As soon as that ATP binds to that head, it will release the actin filament and the contraction will stop. Then you have catabolism here, decomposition of that ATP for ADP and phosphate again. That energy, the energy was all used up in this power stroke, so we need the ATP and decomposition or catabolism to happen so we can store more energy in this myosin head, so you can have another stroke, so you can have another contraction, that power stroke, so you can have another contraction. So the heads of the myosin will bind after the influx of calcium and the active sites are exposed, and then the myosin heads will come in contact with the actin and pull that actin along in that power stroke. This is contraction. As the power stroke is happening, the ADP and phosphate will be released from the myosin head. And then as soon as it's released, an ATP, if it's available, need a lot of ATPs for your muscles to keep working. And so ATP comes in and attaches to the myosin head. As soon as that happens, this bridge is broken, this contact is broken here. That ATP will catabolize into ADP and phosphate. The energy is released. So it is stored in that head. If you have enough calcium and to keep those active sites exposed, you can have another cross bridge, one after the other. So you have to have plenty of ATP and plenty of calcium for muscle contractions to happen, and that's called the power stroke and the sliding filament theory. So when we looked at the parts of a sarcomere, we had the myosin and the actin, we had the I band and the A band, and that A band was the length of the myosin. The A band never changes width or length. The actin itself never changes length in a contraction. They're sliding past each other. So they never change in length. Now, what happens when blood calcium levels drop? You know, the chemoreceptors are monitoring blood calcium levels, and as soon as it's indicated that the blood calcium levels have dropped, hormones are sent to the parathyroid hormone or parathyroid gland, so it will release its hormone, parathyroid hormone. That's picked up by the blood and taken to the bones, and that activates those osteoclasts. And then you have bone resorption, or breaking down of that bone, to release bone calcium into the blood to restore blood calcium levels so that you have enough calcium for muscle contraction and many other things. Now, this is just a review of the structure again. Here's the sarcolemma. This is where you would have an influx of the change. You'd have a change of the polarity of the membrane. An impulse causes the change. The influx of sodium, which changes the polarity of this membrane, and that allows for the influx of calcium into the sarcoplasmic reticulum. All right, here's the cisterna, the sarcoplasmic reticulum, cisterna, the sarcoplasmic reticulum, and the transverse tubule in the middle, and together they're called the triad. This is that transport system of electrolytes that are needed, but one of the main ones is calcium. So the delivery system to the calcium for muscles to aid in muscle contraction.
An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
These animations are found in the PowerPoints in Canvas or as standalones in one of the modules on Canvas for this unit module. Okay, when the sarcomeres shorten, we looked at this on a model in lab, and we talked about this last time. The parts of the sarcomere from a Z-line or Z-disc to a Z-line is going to be a sarcomere. Okay, that's the thick protein filament myosin, and attached to the Z-line are the thin protein filaments of actin. Okay, remember the actin is the one with the troponin and tropomyosin. And before we get any further, we need to say again that these protein filaments do not shorten themselves in a contracted muscle. They do not shorten. The actin doesn't shorten. The myosin does not shorten. When there is a contraction, when the sarcomeres shorten, the thick and thin filaments slide past one another, where you have those myosin heads binding on the binding sites of the actin. When the tropomyosin exposes the binding sites from the calcium binding with the troponin, that causes the tropomyosin to move and expose the active site, the myosin head binds with the actin molecule and pulls it along. You have cross bridging, that's when they touch, and then you have a power stroke where it pulls that actin along. When this happens, then remember these parts. You had the I band here, the A band here, the H zone, the M line. When that power stroke happens, when you have muscle contraction, the H zone becomes much more narrow and may even disappear. The Z lines get closer together, so the sarcomeres shorten. These slide past one another to make a more narrow H zone, and the sarcomere shortens, so the Z line or Z discs become closer together.
In a relaxed muscle, actin and myosin myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins slide toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap, the H zones disappear, and the I band becomes very narrow.
Again, no changing in length of the myosin and actin. In cross-bridging, this is just a review here, the actin and myosin cross-bridge bind. You have the influx of calcium. The calcium binds with the troponin, changes the shape of the tropomyosin, which exposes the binding sites on the actin. It allows for this energy-filled myosin head to bind with the active sites on the myosin. The myosin cross bridge pulls the actin. It's using that stored energy in that head to pull that actin filament and that cross stroke and that power stroke. The ADP and the phosphate will detach, and then an ATP binds to the myosin head, and that causes the breaking of these bonds between the actin and the myosin head. The linkage between the actin and myosin bridge breaks, and then the ATP splits. We have catabolism again. When the more complex ATP becomes broken down, it releases energy in catabolism, so that catabolic energy is stored in that myosin head. Myosin cross bridge goes back to its original position. If you still have the presence of calcium, you would still have active sites exposed. You now have this energized, the energy restored in that myosin head. If everything is just right, you've set your dominoes back up, and you will have another cross bridging and power stroke or muscle contraction.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
Now, we don't want continuous stimulation of our muscles. That would be tetanic contraction, the contraction of that muscle without a release. And so that doesn't happen, we have an enzyme called acetylcholine esterase. Remember, the ASE ending at the end of a word indicates that that is an enzyme. The enzyme acetylcholine esterase helps to break down the acetylcholine into acetic acid and choline. It goes from the synaptic cleft back into the synaptic knob, into the synaptic vesicle, and binds together to form more acetylcholine needed for the next muscle contraction. So if we had a continuous supply of acetylcholine, calcium, and ATP, we could have a continuous muscle contraction, and we don't want that. So our acetylcholine esterase helps to break down the acetylcholine so you don't have continuous stimulus. And when that acetylcholine is broken down, the calcium moves back into the sarcoplasmic reticulum, and the myosin and actin binding is prevented. So we need this for muscle relaxation. And I want to throw a couple other things in here. You need to remember how the nerve poison or nerve gases will inhibit the production of acetylcholine. Now, what does that mean for us? Well, nerve gas inhibit acetylcholine release. Well, acetylcholine has to be present for the influx, the depolarization of the membrane, and the influx of calcium. If we don't have those two things, then our muscles won't contract. And if our muscles are not contracting, what do we call that? Paralysis. What's the most important muscle of our body to stay alive besides your heart? The diaphragm. Okay, so a nerve gas inhibits acetylcholine release, therefore causes paralysis. So a person will die from suffocation because their diaphragm will not work. It's paralyzed. So as long as it takes for you to die from a lack of oxygen is how you die, and it's not a very fun way to die. Anyway, in some weed and fire ant killer, there is a chemical, a poison that causes too much acetylcholine to be released. And so if you ever watch an insect die from too much acetylcholine, it's pretty violent because they have uncontrolled muscle contraction. I believe I'm going to stop part 2 at this point, and we'll start next time with part 3 of the muscular system.